So, have you ever wondered what happens to an amino acid after it drops off its nitrogen? Well, it leaves behind this thing called a carbon skeleton, and our body, being incredibly resourceful, definitely doesn't let that go to waste. Today, we're going to dive into the really fascinating metabolic fates of these carbon backbones.
So yeah, this is the big question, right? After transamination or deamination strips away that amino group, we're left with what's called an alpha keto acid. That's our carbon skeleton. Now, the body is super efficient. It doesn't just toss this aside. Nope. This carbon backbone becomes a really valuable source of metabolic fuel.
Here's the game plan for today. We'll start at this critical fork in the road these carbon skeletons face. Then we'll follow both paths. One leads to glucose, the other to ketones. We're also going to look at what happens when these pathways, break down. And then we'll wrap up with the key takeaways you absolutely need to remember.
Okay, let's get into it. Section 1, a fork in the road. Before we can even talk about the two different destinations, we've got to get to know our traveler, the carbon skeleton itself. Let's define what it is and the fundamental choice it has to make inside our cells.
So, what is a carbon skeleton exactly? Well, it's just the alpha keto acid that's left over after an amino acid is done with its job and its amino group has been removed. The key thing to get here is that this is not waste. It's a super valuable and versatile building block, all ready for its next mission in the body.
And here it is, that fundamental choice I was talking about. Every single carbon skeleton gets classified as either glucogenic, which means it can be turned into glucose, or ketogenic, meaning it's destined to become ketone bodies. This one distinction has massive implications for how our body manages fuel, especially during different states like fasting or exercise.
Now, what's really cool is just how elegantly our metabolism works. I mean, think about it. We have 20 different amino acids, but all their carbon skeletons get funneled into just 7 key molecules. These 7 intermediates are basically the on-ramps to our central metabolic highways, like the TCA cycle.
All right, let's take that first path, the road to glucose. This is the journey of the glucogenic amino acids. They're kind of the unsung heroes responsible for keeping our brain supplied with sugar when our carb stores are running low.
One of the most important gateways for making new glucose is through the molecule pyruvate. A whole family of amino acids, like alanine and serine, get broken down into pyruvate, which creates a direct bridge from protein breakdown right over to maintaining our blood sugar.
Then, you've got another group. They enter that central metabolic highway via a TCA cycle intermediate called alpha-ketoglutarate. Amino acids like glutamate and proline are converted into this molecule, which just reinforces their potential to become glucose.
The branched-chain amino acid valine, along with methionine and a few others, takes a different route. They enter the TCA cycle as succinyl-CoA. This is yet another critical entry point that can ultimately lead to the synthesis of glucose.
And finally, we've got the last two on-ramps. The aromatic amino acids, phenylalanine and tyrosine, can form fumarate, while aspartate and disparagine are really easily converted to oxaloacetate, which, is the direct precursor for gluconeogenesis.
So what's the big takeaway here? Well, it's this. When you're fasting, or maybe you're on a very low-carb diet, your body turns to these glucogenic amino acids. It breaks down protein to harvest their carbon skeletons, making sure your brain and other tissues have a steady supply of glucose to run on.
Okay, so we've seen one path. Now let's double back to that fork in the road and check out the other one, the ketogenic pathway. This route leads to the production of ketone bodies and fatty acids, which is a whole other type of fuel source.
The ketogenic amino acids, they break down into either acetyl-CoA or acetoacetate. Now here's the absolute unbreakable metabolic rule you gotta remember. The reaction to turn acetyl-CoA back into pyruvate, it's a one-way street, and in humans, it's permanently closed. This is exactly why these carbon skeletons can never be used to make a net gain of glucose. It just can't happen.
And this is where it gets really nuanced and interesting. We have 2 amino acids, leucine and lysine, that are purely ketogenic. That's it. Their fate is sealed. But then we've got this really versatile group like isoleucine and phenylalanine that are both glucogenic and ketogenic. Their carbon skeletons can actually be split, with different parts going down each path depending on what the body needs at that moment.
Look, understanding these pathways isn't just for exams. It's so crucial because when they go wrong, the consequences can be incredibly severe. Let's take a look at a few key clinical correlations. First up, we have maple syrup urine disease, or MSUD.
In this condition, a single enzyme complex, the branched-chain alpha-keto acid dehydrogenase, is defective. This causes a massive metabolic traffic jam. You get a buildup of the keto acids from valine, leucine, and isoleucine, which are toxic, and, well, they give urine its characteristic sweet smell.
Next is phenylketonuria, or PKU. Here, the enzyme that's supposed to convert phenylalanine to tyrosine is just missing. So phenylalanine gets shunted down this alternative pathway, creating toxic metabolites like phenylketones. These cause severe brain damage if the condition isn't caught and managed with a special diet right after birth.
And finally, there's homocystinuria. This is a genetic disorder that messes up the metabolism of methionine. A key enzyme is faulty, causing a toxic buildup of a substance called homocysteine. And this stuff just wreaks havoc on the body. It affects everything from connective tissue and vision to the nervous and cardiovascular systems.
So why does all this matter in more common conditions? Well, in states like prolonged fasting or uncontrolled diabetes, the body relies heavily on breaking down protein for fuel. The carbon skeletons literally become the primary feedstock for making both glucose and ketones. So understanding these pathways is absolutely key to understanding the metabolic state of these patients.
All right, let's lock all this in. Time for a final revision. Here are the key takeaways you absolutely need to remember.
So here's the entire story in just 4 steps. Step 1, deamination leaves a bare carbon skeleton. Step 2, that skeleton jumps into the central metabolic grid through one of those 7 key entry points. Step 3, from there it faces its destiny, either become glucose or become ketones. And step 4, as we've just seen, when any step in this elegant process fails, the clinical consequences can be really, really serious.
Look, if you remember only one specific fact from today, make it this. Leucine and lysine are purely ketogenic. That's it. That is the high-yield fact that shows up on exams all the time. And we'll end with a little clinical challenge for you. Based on everything we've just talked about, how would you design a diet for a patient with PKU? What absolutely must be restricted? And just as important, what might need to be supplemented? Answering that, that's how you turn biochemistry into real-world medicine. Thanks for joining me.